Continuing with our neuroanatomy lecture, we are going to move into the meninges. So the meninges are layers that are outside of the brain but under the skull and provide multiple layers of protection and other support for the brain. There are three main layers that we'll talk about. There's the dura mater, which is directly under the skull. There's the arachnoid, which is below the dura mater, and there's the pia mater, which directly encases the brain. So dura mater, you can see here, if you were to do a dissection of the brain, it would be this tough outer tissue. And dura mater means hard mother, so it's the hard protection around the brain. The arachnoid, you can see here is kind of the, it's not quite as hard, more of a matrix that surrounds the brain underneath the dura mater and allows for movement of fluids. And then we have the pia layer, which you can't really see because it is directly on top of the brain and it covers it very well, but it would be under. So you have you can see all of these blood vessels running through the arachnoid layer. So these are important for support and for protection. Some types of brain hemorrhage. So this, I'm a TBI researcher, so this is one of the things we talk about, but it's also useful terminology for understanding the different layers of the meninges. So in this image here, you can see the skull, the dura, the arachnoid, and the pia layer, sort of here, and a few different types of hemorrhage or bleeds that we can talk about after a traumatic brain injury. These are, going from the outside inside, an epidural hematoma, which means that it is under the skull but above the dura mater. There is the subdural hematoma, meaning that it is subdura, it is under the dura mater, but above the arachnoid. We can have a subarachnoid hemorrhage, which would be within the arachnoid space. And we can have an intracerebral hemorrhage, which just means that it's in the brain. There are also intraventricular hemorrhages. Those happen within the ventricles or the fluid-filled spaces in the brain. Here's what some of those hemorrhages look like on MRIs. So here, for example, is an epidural hematoma where the blood shows up very bright in this. Here we have a subdural hematoma, so it's under the dura mater. And here, a subarachnoid hemorrhage. So there are different MRI sequences that we can use to see each of these. Some of them are more sensitive to some types of bleeds and some types of lesions than others. The ventricles. So there's a ventricular system in the brain that is critical for the movement of cerebrospinal fluid, which we'll talk about in a minute. But these are essentially fluid-filled spaces in the brain. So we talked about gray matter and white matter. CSF is the other main tissue type. It's not tissue, but when we are looking at the brain and we're trying to separate out the different segments, CSF is one of those main segments. So there's a ventricular system that goes throughout the brain. Some of the regions that you can see here, both in this diagram and on MRI, we have the lateral ventricles that are on either side, so lateralized. We have the third ventricle, which you can see here in the middle, and the fourth ventricle, which is here behind the brainstem in front of the cerebellum. There are a few other smaller regions that are important for this flow of CSF, but those are the main ones. And CSF, or cerebrospinal fluid, contains lots of nutrients. It's really important for cushioning the brain. Again, coming from the perspective of a TBI researcher, it acts as a cushion. And just like the experiment, if you were to put an egg in a jar and shake it, the egg will break. But if you put it in a jar and fill it with water and shake it, it's less likely to break. This is what one of the important things that the CSF does for us as well. It's also important for removing waste products and just generally helps the central nervous system function. The CSF is created by the choroid plexus, which you can see here. It's these little tissues that are inside the ventricles and create the CSF. So some of the other regions we've talked about generally gray matter and white matter and CSF. And some of the landmarks that you can see, such as the fissures, the certain gyri. What I'm going to talk about now are subcortical gray matter structures. So these are gray matter structures, which we generally can think of gray matter on the outside, but on the inside. And there are a few of these that are important that come up a lot, just across all research, whether it's focused on a particular disorder or focused on a particular function. They serve a lot of different functions. So there is the hippocampus, which you can see here in yellow. And the hippocampus is important for consolidation of memory, spatial memory. And it is one of the first and most highly impacted regions in Alzheimer's disease, which makes sense because it's a disorder of memory. And it's also impacted in many different psychiatric disorders. So it's not really specific to any particular disorder. So it seems like it might be just a generally stress-sensitive structure in the brain. We have the amygdala here in red. The amygdala is important for fear processing, so it's responsible for your fight or flight response, and it is critical for emotional memories. Moving on, we have the putamen in pink here on either side. The putamen is important for movement and learning, and it's one of the structures that's impacted in Parkinson's neuropathology. So Parkinson's is a disorder of movement. So that makes sense that we would see some disruption here. The globus pallidus is another region that's important for movement, and you can see it here in purple, just right next to the putamen. The thalamus, as we talked about on an earlier slide here in green, which is in the center of the brain, the thalamus is a key gateway to the cortex. So it relays sensory and motor signals from the outside world up to the brain where it can process it and back out. And we have the caudate here in teal. The caudate is important for memory and for goal-directed movement. So going in a little bit more detail in the cortical anatomy of the brain, again, understanding that there are individual differences, there are some structures, a number of structures that are represented in some form across most individuals. So we're going to go over what all of these gyri are called now. So in the frontal lobe, we have the superior frontal gyrus, the middle frontal gyrus, and the inferior frontal gyrus. You'll see that for the most part, the names of these gyri make sense with where they are. Within the inferior frontal gyrus, there are three subregions called the pars orbitalis, the pars triangularis, and the pars opercularis. And together, these are called Broca's region. This is an important area for language production. It's called Broca's region because of Dr. Broca, who discovered the fact that these are important for language production. He had a patient who had lesions in this area and could only say the word tan. That was the only word that he could produce. So this was called Broca's aphasia. Some other regions, the precentral gyrus, we've gone over numerous times. This is the primary motor area. We have in the temporal lobe, the superior temporal gyrus, middle temporal gyrus, and inferior temporal gyrus. And in the superior temporal gyrus, towards the back part of it, is an area called Wernicke's area that is important for language processing. Again, just like Broca's area, this was named this way for the doctor who discovered the importance of this region based on a patient who had a lesion there. Precentral, postcentral gyrus. The postcentral gyrus is important for sensory processing. And one way that we can look at the anatomy of the precentral and postcentral gyrus is through a visual representation called a homunculus, or homunculi, plural. So there's a sensory homunculus and a motor homunculus. And these are just representations of generally what part of the precentral and postcentral gyrus and how much area is dedicated to which part of the brain. So here on the sensory homunculus, you'll see that it generally starts in the bottom of the body and works its way up, all of the fingers here, the face and such. And the the amount of space that's dedicated to each of these regions generally corresponds to how sensitive they need to be. So for example, toes, there's not a whole lot that we do with our toes. Of course, they're important for balance and walking, but it's not like we need to have very sensitive responses there. Whereas our fingers, we use them for so many things. We use them for writing, we use them for using tools, so they need to be a lot more sensitive to sensory inputs. So there's a lot more space that's dedicated to senses in the fingers. And similar over here in the motor homunculus, again, different parts of the brain, different parts of the motor cortex dedicated to different regions. Again, this huge amount here dedicated to the face because of all the things that we do with our face, speaking, eating, communicating with each other. So it takes up a lot of space. And here they are in MRI data. So again, precentral, postcentral gyrus, the central sulcus here, the sensory and motor homunculi. And here you can see some fMRI results from a sensory task. So here we have the postcentral gyrus on both sides being activated during a sensory task. Continuing on to other parts of the brain. In the parietal lobe, we have the postcentral gyrus, which, as we just went over, is important for sensory function. We have the supramarginal gyrus that curves around here for spoken and written language processing. The angular gyrus, which also is important for written language processing, but also attention, spatial cognition, a wide number of things. And then in the occipital lobe, we have the cuneus, which is where the most basic visual processing occurs, and the lingual gyrus here, which is part of the word processing system, and also is important for visual memories. So that's what we're covering here on our course, but there are many other more in-depth courses out there that I suggest that you look into. If you do anything related to neuroimaging, if you go on to become a neurologist, knowing your neuroanatomy is really important. And this is one place where you can go into a lot more depth. If you go to this website, it's freely available. They have videos, they have interactive modules. You can look at cross sections. You can see what things look like both on MRIs and in person if you were to do an actual dissection. So it's a really, really nice resource that I suggest you use. Thanks.